Okay, we are pleased to, yeah, that my department chair, I have to let him in. Okay. Okay. Wait,
Do you
I'm
want me
still,
to continue admitting people while you talk?
oh, yes, 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 you may do that.
Okay, good.
We're pleased to have with us Professor Eric Mazur, who is the Volkonsky Professor of Physics and Applied Physics at Harvard University. His research is in the area of nanophotonics. Professor Mazur received his PhD in physics at the University of Leiden in the Netherlands. Following this, he did a postdoc with Nobel laureate Nico Bloembergen, also at Harvard at the time, to learn about nonlinear optical spectroscopy. So afterwards, he was invited to stay as an assistant professor and has been there ever since. He's won numerous awards in both education and research, including the Minerva Prize, the Millikan Medal, the Esther Hoffman Beller Award. He's published over 400 peer-reviewed journals, articles, more than 50 patents, and has started several successful companies such as Scionx, which commercializes black silicon, and Learning Catalytics that uses data analytics to improve learning in the classroom. He's also an innovator in physics education, author of Peer Instruction, and has pioneered techniques in teaching physics in large lecture classes interactively, which has transferred well to other fields of study. So I've been told that we have a hard stop at 4:15 p.m. Central Time. Yeah, that's right, Central Time. So without further ado, I give you Professor Mazur.
Well, thank you so much, Charles. And I regret not being able to visit in person, but hopefully in the not too distant future, I'll be able to visit you. So you may be wondering what a physicist is doing, you know, in giving a talk in a chemistry department about biology. And in some sense, this talk is about a favorite topic of mine, namely serendipity, and how you know, a lot of things that happen in life in general, but even in research, end up being completely serendipitous. It's certainly been a recurring theme in my life. I started out wanting to be an astronomer, and you know, from very young age, and then I enrolled at the University of Leiden, which was the mecca for astronomy back in the 70s, and within 6 weeks, you know, my whole romance was to heaven unraveled, and because the teaching was just about, you know, plugging things into equations. I was totally disillusioned, and I dropped out and switched to physics, only to discover 6 weeks later that physics wasn't that different from astronomy. But I didn't dare to change a second time. And then in graduate school, I thought, you know, I came from a family of academics. My father was a physicist, my mother an art historian. And I said, I'm going to do something useful in my life. I'm never, ever going to be a professor. I'm going to go into industry. And I got a job in industry, and then I accepted the job. But then my father said, wait, why don't you first take a postdoc in the United States? I grew up in the Netherlands, as you heard. And I thought, well, I've never been to the United States, so maybe that's not a bad idea. So I called my employer. I said, it's okay if I defer coming for a year. And they said, sure, by all means. So I wrote to Harvard, Stanford, Berkeley, and MIT. I thought I have nothing to lose, so I might as well write to those 4 places. They'll say no. I just go to my dream job. And as you heard, I got an offer from Harvard and accepted it. And what can I tell you? It's been a long year. So my life has many 90-degree turns. And in a sense, the story that I'm going to tell you today is a little bit like that too. So let me actually share my my slides with you, so we can begin here. So essentially back in the, when was it? In the, I think late 90s, I was doing research in my group where we were taking femtosecond laser pulses, ultra short laser pulses, and focusing those inside the bulk of a transparent material, right? So the intensity is maximum, not at the surface, but in the bulk. And we discovered that this very high intensity at the focus would actually create a little spark due to nonlinear ionization, and that would actually create, through a Coulomb explosion, microscopic damage in the material. If you would take one of these pieces of glass and put them under the microscope, you'd see these tiny little cavities inside the material. Actually, interestingly enough, and this shows how sometimes it's good not to be completely aware of what happens around you. At the precise moment that we did this, a German group had tried exactly the same thing, and they had published it's impossible to create damage inside a piece of glass by focusing a femtosecond laser pulse. I mean, the opposite of what we found. I think if I had read that paper before embarking on this, I might have just not done it. But anyway, luckily I had not seen that paper. In fact, it didn't come out until our paper came out too, so I could not have seen it. So essentially, the important thing to realize, I mean, this isn't a piece of glass, right? So it's transparent. So the energy of the photon is less than, the transition, the minimum transition, the gap in glass, and therefore it can't be linear absorption. It has to be a nonlinear interaction. And that nonlinear interaction, which will play an important role in the rest of this talk, is what actually provides this bulk confinement, right? If you had linear absorption, then essentially the absorption would start at the surface here, and then diminish as you go into the bulk. But if it's nonlinear, then most of the intensity and therefore most of the absorption will be at the focus. So in a sense, you get 3D confinement of the energy, and that can be really confined to a very small space. In fact, scanning electron microscopy and atomic force microscopy pictures revealed that the cavities that we created were on the order of about 100 nanometers, so less than the width of the light at the focus due to this nonlinear interaction. And even more interesting, because the energy was so low, right? I mean, it's highly confined, so the fluence, the intensity is really high, the energy per unit area, but the total amount of energy, as you will see in a moment, is actually very small. And therefore, around the places that are irradiated, there's absolutely no damage. As soon as the energy dissipates out of the focus here, it sort of diffuses and dissipates in 3 dimensions and creates no damage. So it's extremely localized. In fact, let me show you some graphs here, and then you'll see the connection to biology in just a second. So one of the things we discovered was that the threshold energy, the amount of energy you need, is a very steep function of the focusing. At very tight focusing, you need very little energy. And if you take so with microscope objectives, for example, if you take ordinary lenses, it very quickly goes up tremendously. And that actually explains why the German group was unable to see anything, because they were actually using an ordinary lens to focus rather than a microscope objective. So in a sense, it was a stroke of luck that they had chosen a lens and we had chosen a microscope objective. So the important point to remember is that let me forward here. Why is it not forwarding? There we go. So at very high NA, this works at numerical aperture. This process works best. And here's really something interesting. It's nearly material independent. Whether you take sapphire or glass or a polymer or even a liquid like water, and of course you need to be away from an absorption, otherwise it's linear rather than nonlinear. It is nearly material independent. It may change by a factor of 2 or so, but not much more than that. So here's another point I want you to look at. at the highest numerical aperture, so 1.0, 1.5, so these are oil immersion microscope objectives, the total amount of energy is just a few nanojoules. A few nanojoules, right? I mean, if you eat a candy bar, it's 100 kilojoules. Or you drop your keys on the floor, it's again, you know, it's maybe a joule or so, depending on how many keys you have in your pocket. A nanojoule, I had to think a long time. What is a nanojoule? Well, imagine a fruit fly flying and bumping into your cheek. I mean, I'm not talking about a fruit fly splattering on the windshield of your car when you're going 55 miles an hour. No, a fruit fly just flying through space and hitting your cheek. That would be about a nanojoule. And you can actually the solid with that amount of energy, provided you deliver that energy over an extremely short amount of time, and you deliver it in an extremely small volume of space, less than a cubic micron. So through this research, I'm not going to tell you more than this about, we found out that, you know, in transparent media, you can induce nonlinear interactions that disrupt matter inside the bulk rather than at the surface. And you can get this complete disruption of even a solid material at very, very low energy, nanojoules. And you have absolutely exquisite spatial confinement. Well, it turns out that roughly at that same time, we were planning our nanoscale manufacturing facility. It's beautiful. If you ever come to visit, I'd love to show it off to you. It's a fabrication facility of, you know, absolutely state of the art. Anyway, we were planning it, and in order to get faculty across the university, across disciplines too, interested in this new investment, we had a series of talks where they would invite different people, biologists, chemists, physicists, you name it, to discuss problems in their research at the nanoscale. And I remember that at one of those talks was a colleague of mine from Harvard Medical School, Don Ingber, who is interested in studying viscoelastic, among other things at that time. He was interested in studying viscoelastic properties of actin fibers in cells. And he developed this tensegrity model. You may have seen the tensegrity structures, which consist of tubes and steel wires. And even though the steel wires are flexible and soft, the whole structure is rigid. And he sort of applied that model to cells to explain, you know, locomotion of cells, signal processing in cells, and so on. Anyway, he was giving a talk on a subject that I didn't know anything about. And one of the things at the conclusion of his talk was that he said that he would be really interested in measuring the viscoelastic properties of these actin fibers. And yes, you can take one of these fiber bundles out of a cell and then sort of measure the elastic properties, but then it's not in its natural environment, and it's not affected by all of the biochemistry that takes place in the cell. Anyway, I thought about it and I thought, boy, I think I have the perfect tool for that, right? Because we could go in with our femtosecond laser pulse, and then we could just cut one of these fiber bundles and then look at the retraction. Anyway, I had no idea what I was talking about or even thinking about, so I was too shy to even say anything. And but after the talk, I went to him and I said, you know, I think we have a tool that could actually help address your question. And that started me on a journey in biology that goes on to this very day. So I'll give you, you know, rather than going in depth anything, you know, it's the end of a Friday, we're all tired, right? So I'm going to give you a broad overview of some of the things that we've done, going back to those early days and around 20 years ago to now. And I think there's an interesting lesson, as I said, in serendipity and in how research can meander around. So I'll talk a little about subcellular surgery, which started with Don Ingber because he was interested in these actin fiber bundles in cells, and then we'll talk about nanosurgery, and then finally about what we're doing now with intracellular delivery. So essentially, after I spoke to him and he got modestly enthusiastic about our capabilities, the first question we want to settle is, can we actually ablate material on the subcellular scale? Is it possible for us to cut something inside a cell? And we decided to start actually by looking at the nuclei of cell. They're easy to see under the microscope. And so we took fixed, not alive, but fixed endothelial cells. And under fluorescence microscopy, what we did is we essentially irradiated lines by, it's a pulsed laser. So we scanned the cell while the laser fires its pulses, varying the energy. So you can sort of see here a line and another one there and another one there. These were done at 3 different energy per pulse regimes, 1.5, 1.8, and 2.3 nanojoules. But if you see it getting darker, it could mean many things, right? It could mean that you have actually ablated material, but it could also mean that you've just killed the molecule that fluoresces. You cause bleaching, in which case it's not interesting at all because the material is still there. There's just no fluorescence anymore. So in collaboration with UMass Medical School, we did actually stereo electron microscopy. So for electrons, if you ablate material, it gets brighter because the electrons go through more easily. And as you can see, at 1.8 and 2.3 nanojoules, you actually do remove material in the nucleus. But at 1.5, you can see absolutely nothing. So at 1.5, we get bleaching, but not ablation. At 1.8, we get ablation. At 2.3 nanojoules, we can get ablation. So that permitted us to make a graph where we look at the widths. We look at the widths of these trenches, if you want. So we put the widths here versus pulse energy, and we combine our transmission electron microscopy images like this, and the data obtained from the fluorescence microscopy in one graph. At very low energy, at the lowest energy that I showed to you here, we can see nothing in TEM, no removal of material, but we see a bleached trench in fluorescence. And then at higher energy, we see the 2 coincide. At lower energy, we see nothing at all. So that means that there are 3 regimes. There's a regime where there's no damage at all. There's a regime where there's ablation. And then there's in between this regime where there's bleaching. And one of the things that we established, and this turned out to be really, really handy in our research, is that typically the threshold for ablation is about 25% higher than the threshold for bleaching. Well, because you can't do these measurements are very time consuming, and then you have to go to this to transport your cell from the fluorescence microscope at Harvard to the electron microscope at the UMass Medical School in Worcester. So and you have to keep the cell in the right position. You have to know where it is on your slide. So it's extremely complicated. But knowing that the ablation threshold is 25% higher than the bleaching threshold, regardless of what type of material you're in, makes it possible for you to essentially slowly increase the energy until you see the onset of bleaching, and then you crank the intensity up another 25%, and you can be pretty sure that you are actually removing material. So that permitted us to actually move forward and to start looking at live cells. So the next experiment that we did was still with endothelial cells, but to look actually not at actin fibers, but at mitochondria. I don't know if you've ever looked at a biology book, but mitochondria look like these perfectly bean-shaped organisms. It looks nothing like that in a real cell. It's like spaghetti, as you can see. And so that was very surprising to me, but that shows how little I knew and still know about biology. And we thought, maybe what we can do is actually remove or cut one mitochondrion. And there was actually a whole, not controversy, but disagreement among cell biologists about whether mitochondria form a network or are essentially isolated individual organisms. So notice here that we have 3 mitochondria that are very close together. I think there's something like yeah, like only about half a micron or so apart, 500 separated by 500 nanometers. So we can focus our laser beam right here where my cursor is, and lo and behold, look at that. We have removed one mitochondrion right here without affecting the neighboring mitochondria. And by doing sort of comparative imaging, we could actually show that they're not actually, they don't form a connected network. I'm going to show you a little movie to show this in real time. So we're going to actually take this mitochondrion here and irradiate it, and look what happens. This is real time. I think the laser beam was right here. It pearls up and disintegrates into four. Here you see it closer up there. So this is about real time. We irradiate. You don't see the laser pulse because it's infrared, and the camera that recorded this doesn't see in the infrared. So at this point, we were really ready to tackle the question that he was really interested in. Can we actually probe the dynamics of the cytoskeleton? The cytoskeleton is formed by these actin fibers and by microtubules too, but we were interested in the actin fibers. So we took a live cell and tagged with yellow fluorescent protein. So all of these fibers actually express yellow fluorescent protein, which is why you can see them. And then we aim our laser at one of these fiber bundles, and boom, it just cuts the fiber bundle, which then actually retracts over time, just 10 seconds later, as it retracts. But does it really retract, right? These actin fibers are complicated polymer molecules. Maybe what is happening is simply that the ends of the fibers depolymerize, and that there is no elasticity in these fibers, and that what we're actually measuring by looking at the retraction is not a viscoelastic retraction, but simply depolymerization. So this is again where the bleaching came in really handy, because what we decided to do was create a bleached marker. So this little dark spot that you see here at the arrow is below the ablation threshold, but above the bleaching threshold. So here we've simply lost the fluorescence, but we've not lost any material. And then we move the laser sideways along the fiber. We crank up the power by about 50%, so that we're sure that we've cut it. If it's depolymerization that's going to happen here, then this marker should stay at the same spot. If it's viscoelastic, then as this end retracts, the little marker should also move sideways. And lo and behold, as you can see here, it neatly moves sideways, showing that it's indeed an elastic retraction. I had a really nice movie showing the retraction with time, but I couldn't find it. I have to find a way of locating it. But I showed you a few time frames. So that made it possible to look at the retraction distance as a function of time, and you can fit that with an overdamped spring, so that you can get a time constant out of this curve. And this is when, of course, my colleague Don Ingber got really interested, because now what you can do is you can introduce in the cell a myosin inhibitor and see if the myosin inhibitor affects the viscoelastic properties of the actin fiber. And sure enough, it does. And different myosin inhibitors have different effects. So now all of a sudden you can test different inhibitors on the cell and the effect that they have on the biochemistry of the cell. In fact, he got so excited that he decided to buy his own femtosecond laser. And all of a sudden I realized, here I am with a laser and no biology and no problem. It's actually dangerous for me as a physicist to embark on biology, because even if I contribute a technique or a tool, if the biologists get too excited about it, I'm left with just a tool and no problem to solve. So just as I was pondering whether to continue with the research that we had embarked on, or just go back to my solid state materials like glasses, there were plenty of interesting problems to tackle there. a serendipitous event happened, just as, in fact, the meeting with Don Ingber was serendipitous. We were interviewing a junior faculty for a junior faculty position, and one of the applicants gave a talk on a little nematode, C. elegans. I'm going to show a picture of C. elegans in just a second, because he was interested in probing the neurological origins of behavior. So C. elegans is a small worm that is about 1 millimeter long and about the width of a hair, and it's almost transparent, which is important. That's a recurrent theme. A cell is almost transparent. C. elegans is also almost transparent. And what is interesting about C. elegans, which is one of the model organisms in biology, is it has about 1,000 cells. So it's a little bit more complicated than the cells, the individual cells that we studied so far. But of those 1,000 cells, 302 are neurons, not 301, not 303, but it's 302. And they have an invariant wiring diagram. So in other words, if you place two C. elegans next to each other and you analyze it, the 302 neurons are all wired the same way, which is very different from our brains, which of course have many more neurons, which are wired in different ways. These are all wired in exactly the same day. It's almost like in the same way. It's almost like, Toyota running off the assembly line where the wires in the car are all wired the same way. And in spite of this simplicity, this neuronal system of 302 neurons completely encodes the behavior. They can detect chemicals. They can detect temperature exquisitely, actually. If you put them on a plate where they eat E. coli bacteria, where you have a temperature gradient, and you put the E. coli bacteria at 21 point, at the place where the temperature is 21.5 centigrade, they'll learn that at 21.5 centigrade is E. coli, and if you then remove the E. coli, you keep that temperature gradient, or you change the temperature gradient, they'll crawl back to the place where it's 21.5 within a few tenths of a degree. So not only can they detect, they can learn, and they can remember with just 302 neurons. So he was studying these behavior by doing an assay by looking for mutants. Is there a mutant that misses this particular neuron? What is the effect of missing that particular neuron on the behavior of the animal? And I was listening to that, and all of a sudden I had the same sensation as I had when I listened to Don Ingber. I thought, first of all, what if you can't find the mutation you really want to find? And wouldn't it be a lot easier if you came in with a laser and disabled a particular neuron? That way, at least you know that there's nothing else different in the animal that has been caused by this mutation. Plus, you don't have to kill the whole neuron. You could just kill an axon or a dendrite and a connection between neurons. So I went to him after his talk and I said, maybe I have the right tool for you. Maybe we can use femtosecond ablation to, instead of looking for a mutation, which you might not even be able to find, to look for, to dissect the neuron. Anyway, long story short, we hired him. He became a junior faculty in the physics department at Harvard. And even before we took him on, we were already doing experiments. So the first neurons we looked at were the ASH neuron. All of the neurons, by the way, it's really amazing to get into C. elegans. If you don't know about C. elegans, you should look up Worm Atlas. Since you're at your computer, it's really easy to do. There's a parts list for, it's really like a car. I mean, every single cell has been identified, and almost every cell has been identified and labeled. I mean, so you can actually scan through the worm and identify each and every little neuron. Now, the ASH neurons are responsible for chemical sensing, which is important, right? I mean, you're a little worm, you're underground, you don't want to kill yourself by having something, by ingesting something that is toxic. And essentially, the neurons are sort of in the brain, primitive, if you want, primitive brain of the little worm. And then there are two dendrites which go all the way towards the mouth of the animal in front. And that sort of extends through the skin at the mouth. And that makes sense, right? Because you want to have advance warning. You don't want to start eating something and then later discover that it's not something you want to eat. And there's one on each side, very much like, we have 2 ears and two eyes. So here you actually see the neuron, and here are the 2 dendrites, and here's an axon ring that actually connects to another set of neurons that is right here. So this is another little movie. I have it separately, but I didn't put it in this presentation. So if you take a Petri dish and you put these little worms, again, they're less than 1 millimeter long, and they're about the width of a hair. If you put a glycerol ring, so you just put a little bit of glycerol on the agarose on which they crawl, you'll see them crawl around, and as they get close to the glycerol, they go back. Or if you were to take something like a little piece of wood or a toothpick and you tap their nose, they crawl back. So they can sense the pressure, and they can sense, they can do chemical sensing. So what we actually did is we did surgery on those worms by fluorescently tagging that ASH neuron, and cutting the dendrite, and then looking at how many of the worms escape this glycerol ring, how many actually go through this, and if when we prepare the worms for surgery, there's always some where we end up not doing any surgery because something goes wrong. We put them in the wrong orientation under the microscope, and we can't get to the neuron well. So we always end up with a number of worms that have gone through exactly the same preparation, but who don't actually have surgery. We call that mock surgery. And if you look at the number of worms that escapes this glycerol ring after mock surgery, which has not really taken place, we see that none escape. If we look at the mutant that misses the ASH neuron, we find that about 80% escape, and that's of course time independent. The great thing with the surgery is we can do the surgery and see if there's any healing of the surgery. If you do one neuron, it's the same as having 2. So it's a redundant system. They don't need both neurons. If we take 2 neurons, then you can see that the escape rate is about the same, and there's no recovery. So there's no regrowth of the dendrite that goes towards the front of the animal. So after doing this, we went to a neuron that's really near the AFD neuron called the ASH neuron, namely the AFD neuron, which is responsible for temperature sensing. And here there's something from a physicist's point of view that is really peculiar. Again, there are 2 ciliary projections that go all the way to the front of the animal, and one on each side. One on each side to have a redundant system that makes sense. But the ciliary projections are kind of weird, right? Because when you're so small, 1 millimeter by 80 micron, there's really no need to detect the temperature on the outside, right? If you're in a house, you want to know what the temperature is outside, you better put your probe outside, because the temperature inside is going to be different than the temperature outside. But when you're the size of C. elegans, the temperature's in equilibrium under all circumstances between a few microns away from the skin and at the skin. So what is the reason for those ciliary projections? So what we decided to do was actually do a number of surgeries where we cut at different places along the dendrite to see where the temperature sensation happened. Does it happen in the neuron body, or does it happen at the very tip of the dendrite? And that's something you cannot answer by just looking at mutants. So the type of experiment that my colleague R.V. Samuel did was an assay where essentially, by the way, here are the AFD neurons, and this is the axon ring for the AFD neurons. And you can see here again a cut in the projection that goes towards the nose. So we essentially move this around to find out where the sensation of temperature took place. And we developed a microdroplet assay where we put the worms in a little glass slide, a suspended droplet from a glass slide. And essentially, we put the worm in the droplet, and essentially they can swim straight forever because they swim against gravity on the side of the droplet. And as I said, the worm has a very exquisite sensation to temperature. They can find where food was in the past down to a few tenths of a degree centigrade. So if you vary the temperature of the droplet over time sinusoidally, what you'll find is that they'll swim, and then if it gets too hot to their taste, they take a turn. And then if it gets too cold, they'll take a turn again. So you can sort of sinusoidally vary the temperature and measure the number of turns they take. So you illuminate the droplet from the bottom and look at it from the top with a microscope, and you can essentially measure the number of turns they take as a function of the variation in temperature to see if they still have that temperature sensation or not. I'm going to show you, because it's a rather subtle effect, I'm going to show you before and rather extreme before and after surgery effects. So here you can see one that is just swimming, and they keep on swimming straight at steady temperature. If you change the temperature, you'll see them turn. And when they turn, you actually see the tip, which is on the left, come very close to the tail. Now, as I said, the effect is relatively subtle, and you have to accumulate a lot of time data, which would not be possible in this talk. So I'm going to actually show you a surgery that went wrong, where we actually damaged much more than the AFD neuron. But you can see that the behavior is very, very different in this case. So if you're interested in the details, I'm going to give you the result in just a second. I can refer you to one of our papers, and you can actually see the data, not visually, but you can see the data plotted as graphs. But the one thing that became clear was that the temperature sensing occurs at the very tip of the dendrite. And as I said before, there is no physical reason for that. So there must be some kind of evolutionary basis for the fact that the temperature sensation occurs at the very tip of the dendrite, and that in a sense, the sensation of temperature parallels the sensation of taste, if you want, the chemical sensation. Anyway, that was a success again for femtosecond ablation in biology. And my junior colleague, who in the meantime had been promoted to associate professor, got really enthusiastic, and he decided to buy his own laser. And all of a sudden there I was again, with an experiment, and the biological problem walked away from me. And I thought, it's time for me just to go back and do physics rather than devote part of my effort to biology. And just as I was completely, decided that I was not going to do any biology anymore, another colleague of mine knocked on the door and said, "Hey, I read about the work you did with Don Ingber and Arvi, and I'm very interested in the spindle that develops during mitosis. And do you think we could?" Anyway, the whole story repeated again, and he too bought a laser after a couple of years. I'm not joking. I see people laugh, but it's a true story. So, that did cause some soul searching, and but then I thought, oh, pardon me, I stopped sharing. That's not what I wanted to do. And that actually optimized for video clip. I don't know. Did the video clips play all right? Because I'd forgotten to hit the
yes, it went, yeah.
Okay, I'd forgotten to hit the optimize for, I'd forgotten to hit the optimize playing video button. Okay, so at the same time, you heard Charles mention at the beginning black silicon, which in itself was a totally serendipitous discovery in my lab. But the surface of black silicon looks like a bed of nails. The surface of silicon develops all these conical structures that have big asperities. And whenever there's a very sharp asperity, you have a huge field enhancement, right? I mean, lightning rods. Just think of lightning rods. And so we became very interested in doing surface enhanced Raman spectroscopy, for example, right? Because you could strongly enhance the intensity of any plasmonic resonance at the tip of these peaks. Silicon is a semiconductor, so you have to coat it with a metal in order to do it. So that led from black silicon to surface enhanced Raman spectroscopy. And I had a postdoc in my group at the time. We thought, you know what, maybe you can use this as a way of delivering cargo to cells. You have this high intensity, you put a cell on it, it porates the membrane, and you can get chemistry in it. A lot of people, after I talk about subcellular surgery, ask me, when can we use this to treat diseases? And I have to remind people, look, we consist of 150 trillion cells, so poking a hole in one cell is not gonna do anything. In fact, whenever you pass your hand through your hair, you shed thousands of cells. So it's nothing that could ever really help on our scale. But if you can do intracellular delivery and you can deliver cargo, like gene editing cargo, for example, to cells, then it would become possibly very interesting. So the question that all of a sudden came to mind here is, can we use lasers for new therapies? Can we cure people, for example, from AIDS? That's why I'm showing a picture of, he just passed away. I forgot his name now. The Berlin patient, the first person to ever get cured of AIDS. So the idea is this, you can take stem cells from bone marrow. I don't know if you know the story of the Berlin patient. It's an interesting story. He got diagnosed with AIDS in the early 90s. There were no therapies for HIV at that time. And then I think about 10 years later, in the early 2000s, he also had leukemia. And they decided to do a bone marrow transplant. And then after the bone marrow transplant, they suddenly discovered that he was completely cured of HIV, because the bone marrow cells that he had received had a mutation that made him immune, or made his T cells resistant to the HIV virus. So the idea that we had was, imagine you take stem cells and then you gene edit them to have the desired change, and then you transplant them back. The first and the last step are well under control. The middle step is a problem. How can you efficiently deliver cargo to cells for gene editing, for example? So what would be the goal in order to make that really into a viable therapy? You want, one, high variability, right? You don't want to kill the cells while you gene edit them. You want high efficiency. You probably need about 150 million cells in order to have a reasonable chance at a therapy. So if you take one laser and you poke a hole and then in a cell, and then you take another cell and you poke a hole and another one, and you have to multiply it with 150 million, you're never going to get there. So you need the efficiency really measures how much of the cargo goes inside. So that's one thing. The throughput is what I was just talking about, needs to be high too. You need to be able to do, millions of cells in a reasonably short amount of time. And you need to be able to do it with a large number of different cells. So biologists have been studying this. In fact, I was speaking last year at world congresses on poration, and there are many different techniques, from just applying naked DNA to viral transfection. Right now, one of the most often used techniques is electroporation, and H means high, L means low, M means medium. So you see that all of these have some drawbacks, at least in at least one category. There's also laser poration, which we had already shown. We can go in a cell. We can also go to the membrane of the cell and poke a hole in the membrane of the cell. But as I said, the throughput is agonizingly low. I mean, so as to be completely useless. So the idea was to actually use plasmonics at the tip of sharp spikes to get high viability, high efficiency, high throughput, high versatility. So we soon discovered that using black silicon was not the most efficient way. It was not a very reproducible technique. So we decided to nanofabricate, little metallic spikes on a polymer substrate, and then we can put a cell on top of it. We can irradiate the whole thing, and we create high intensity at these peaks, which pokes holes in the membrane of the cell. And then if you have some cargo diluted in the medium, then some of it will diffuse into the cell. So essentially, we start with silicon. We put a photoresist on there, and then pattern the photoresist with an electron beam, and then we actually used light. This was not done with an electron beam. Pardon me. And then we use an anisotropic etch to etch away the silicon. We put gold. We deposit a layer of gold, and then we press on top of that a cover slip with a polymer. And after hardening the polymer, we essentially strip off the pyramid substrate. That's how we fabricate our sample. We can do this in very large quantities. So these are literally, I don't know, tens of thousands of pyramids on each side. You can see it's a little bit darker because the gold layer is not completely flat here. I'll zoom in. By the way, I tried to get this image published, and apparently you're not allowed to publish pictures of quarters or any money. It was rejected, this image. We had to retake it with something else. I'm not allowed to put a quarter in there. That's surprised me. Here you see an electron microscopy image of these little pyramids. They're 2.4 micron on a side, 1.4 micron high, and they're spaced by about 4 microns. Here's a picture of cells that have been grown on top of these pyramids. They're fixed, otherwise you can't do electron microscopy, which is why they look so weird. But you can see how they're actually sitting on top of these pyramids. And then we did our proof of principle experiment. So here you see a substrate that has no pyramids here, but it has pyramids there. You can see them, the little dots. And then we scanned the laser on the top part of this plot of this image, but we did not scan the laser here. You can see cells everywhere, but you can see that only the cells where the scanning took place and where there are pyramids are actually expressing this fluorescent cargo dye, actually, that we put in the medium. So it actually shows that the pyramids permit you to porate the cell, and anything that's water soluble will actually, provided it's not too big, will diffuse into the cell. And just as we were really, really excited, our femtosecond laser broke. We'd been using femtosecond laser pulse. In fact, everything I've told you is femtosecond laser pulse, and everything hinges on this nonlinear interaction that I've been doing at Harvard for close to 40 years right now. And so the laser broke, and one of my students said, we have a nanosecond laser. Why don't we use that? And I said, it's not going to work. Femtosecond to nanosecond, that's a factor of 1,000 in time duration, right? And as you stretch the pulse, the intensity comes down. So we won't have the intensity anymore. And if it's let's say an I to the third effect, a third order nonlinear effect, bringing your intensity down by a factor of 1,000 is 3 times 3 is 9 orders of magnitude in, no, sorry, by a factor of 1,000,000, right? Femtosecond to nanosecond. So that's 10 to the 6 to the third. So that's 10 to the 18. So it's 18 order of magnitude lower in intensity. So I said, no, it's not going to work. Well, anyway, my students decided to ignore me and try it anyway. And you know what? It worked. I mean, I was totally flabbergasted. I mean, we had this idea that it was the intensity on the spikes. That was the whole original idea. And I'd worked hard on trying to dissuade my students. And clearly we had to go back to the drawing board and think about what is going on. What is the mechanism here? We thought plasmons give a near field enhancement, just as in surface enhanced Raman spectroscopy, and that yields to poration because this intense field disrupts the membrane, the lipid membrane of the cell. But clearly that cannot be the case. Well, it turns out the poration is actually mediated by microbubbles. At the tip of these pyramids, you have a plasmonic resonance that creates a high temperature. The high temperature creates a bubble because the water vaporizes, and that bubble actually is what is disrupting the cell. So again, what we want is high viability, high efficiency, high throughput, and high versatility. So this is what we do. We have our substrates. We seed HeLa cells, for example, on the surface. We add calcine green. That's what we want to get in the cell. We scan the laser. Then we add a viability indicator, something that shows whether the cell is alive or not alive. And then we look in both fluorescence microscopy and also use flow cytometry to actually count cells. So here are the pyramids. Here it's flat. This is an electron microscopy image, so it looks darker because the electrons are scattered away. And if you look really, really closely, but Zoom will probably not transmit that, you can actually see the rows of pyramids. We scan the bottom, we don't scan the top. And this is done with nanosecond pulses. And lo and behold, the cells in the quadrant where there are pyramids and where the laser irradiated show uptake of the calcine green. Now we can vary the intensity of the laser pulses, right? I mean, if we just crank up the intensity of the laser pulse more, at some point we just kill the cell. So what is the optimum fluence, energy per unit area of the cell? So here we show the efficiency in black and the viability in white as a function of fluence. As you can see, let's start with the viability. The viability goes down, and that makes sense, right? Because the higher the fluence, the more energy, the more cells we kill. The viability goes up because higher energy pulses make bigger holes in cells, and therefore the uptake is bigger, but it can never exceed the viability. So ultimately it comes back down as we kill cells. And you can sort of see that the optimum range is around, 5 or so joule per square meter. So let's see, which of these can we take off? Well, one, viability. We're close to 100% viability at the optimum frequency, something like 90% or so. And also efficiency, because we got almost as high in efficiency. So we tick off those first two boxes. It also turns out that we can do about 1 million cells in a minute, and this is without any engineering, right? We just scan the cell, it takes 2 seconds with our pretty primitive setup to do 1 million cells in a minute. So the throughput is good too. What about versatility? Well, we want to be able to deliver different cargo sizes. We don't want to deliver calcine green. That's never going to help with any therapy. We'd like to do something like CRISPR-Cas9 and we want to have different cell types. So the first thing we did was look at the size of the cargo. So molecular weight is here on the horizontal, and we looked at both viability and efficiency as a function of molecular weight. Efficiency is not affected by the cargo. That's not surprising because that's affected by the laser pulse, not by the cargo. But as you increase the size, the amount of cargo that diffuses into the cell diminishes. The good news is that CRISPR-Cas9 lies still in a region where the viability is relatively high, around 66 or so What about different cells? Well, we had mostly looked at adherent cells like HeLa cells, muscle cells, breast cells. And all of them are fine, but stem cells, for example, they are not adherent. They won't go down onto a little plate. So we looked at a number of different suspension cells like bone marrow, stem cells, and immune cells we haven't done yet. And in fact, even two primary lines, primary muscle cells and primary stem cells, and we can deliver to all of them. So it's a pretty versatile technique, actually. So I've spoken a few minutes longer than I wanted, but I hope I've convinced you of two things. One is it's a great tool for manipulating the machinery of life, not only to study different materials. You know, I've shown it to you how you can use it to study cell structure and mechanics, how you can study the neurobiological basis of behavior, and how you might possibly open the door for new therapies. And the other thing is, you know, how incredibly serendipitous this journey was for me. And I think that I could never have predicted, I could never have written a proposal about it. And if it weren't for a number of completely serendipitous events in my life, I wouldn't be here now talking to you about it. So I'm the only one speaking here, but you know, there's a large group that has helped me with all of these experiments and funding from a number of different sources. And I want to thank in particular my colleagues Don Ingber at Harvard Medical School, and R.V. Samuel in the physics department, and Dan Needleman in molecular and chemical biology for being such wonderful, and Elena Aikawa at the Harvard Medical School for being such wonderful collaborators. And I want to thank you for your attention.
Well, thank you, Eric, for that presentation. So if we have any questions from the audience, you feel free to unmute yourself and ask Eric. Any?
Okay, I was going to wait, but so with your previous colleague, have you, with the C. elegans, have you or they that you know of tried any of these methods with lines that were deficient in some manner, where you could actually deliver some sort of therapy in a living worm and see the effects? Like, basically I'm asking beyond just a single cell level or even just a plate of even 1 million cells. Have you looked at this in terms of actual organism?
So my colleague R.V. Samuel is still very active, except that he's moved on to different organisms. He's particularly interested in vision and fruit flies. And I have to say, I haven't followed exactly what he's doing right now. And he has added a number of other techniques to his arsenal. My graduate student who started all of this research is now a professor at Northeastern, and he's still interested in looking at C. elegans, at you know, healing of damage to neurons, because that has a lot of importance in medicine in general. And one of the postdocs of my colleague, who is a professor at University of San Francisco, is actually also still working in this area. I'm no longer looking at C. elegans, so I'm essentially I'm out of it, which is fine. I mean, I'm perfectly okay with that. And it's great to see that others have continued. So I don't think that anybody has developed a therapy or anything. It's still mostly used as a research tool. And it is certainly shedding light on biological processes. You know, anything from, oh, I love that picture of a mitochondria that I see behind Neil there, of the, it's wonderful. Is that a coincidence, or did you actually put it up?
It was actually, I put it up during my cell metabolism lecture class, and I just hadn't changed it. But I do have a question for you. I was wondering, Well, first off, it was a great lecture, and I was wondering, when you're talking about delivering payloads such as CRISPR-Cas9, if you or any of your collaborators did any follow-up work to look at whether this, the heat generated by these plasmons, plasmonic resonance damages the enzyme payloads in any way, or what their efficiency is, how they survive through that.
Yeah. So actually, that's exactly where we are right now. The pandemic has disrupted, unfortunately, our research, but I hope to have an answer to that very quickly. So we're collaborating with a number of different groups to actually deliver real gene editing cargo rather than a sugar molecule that has the size of CRISPR-Cas9, and seeing if we can do something. The cell is definitely viable. They continue to divide and so on. So that we know for sure. But whether we can actually do gene editing, I wouldn't see why not, but we have to prove it before people will believe us.
Right. Yeah, I would think that the cells, since they're viable and they can repair their own membranes, but I guess CRISPR-Cas9 couldn't be necessarily repaired by the cell's machinery. So I guess, yeah, I'll be interested to see what you all find out.
Well, no, hopefully soon.
All right. Are there any other questions from the audience? Okay. All right. Well, thank you, Eric, for presenting. Just to let you know, we're past the 4:15 mark, but I was wondering, could you, maybe we can invite you back to share with us what you learned about how to teach during the pandemic, or what things you've learned.
I'd be happy to do that. In fact, I am starting a series of webinars, which are, I decided, if I explain everything I do, people get overwhelmed. So I pick certain aspects of it. So I'm actually starting a series of webinars focused on how to prepare students. So we developed a platform called Perusal, which is,
yes.
Yeah. So I'm actually starting. I don't know when the first webinar is, but I can send that to you, Charles.
Sure.
All
right.
And I'm actually also giving one at Vanderbilt, a whole webinar about it. But I'd be happy to do one for
Sure.
Middle State University. I'd be
Middle
Tennessee State University. I'd be more than happy to do that.
Yes. Well, great.
Okay, so let's correspond about that.
Yes. We'll talk further.
Great.
Yeah. Well, thank you again, Eric. And
thank you. It's fun to talk about bio. Oh, Siri is thinking I'm talking to her here. My iPad started responding to me. I'm not sure. So thank you so much.
All right. Well,
for having me.
Thank you, and everyone have a good weekend.
See you. Bye-bye.